Hello friends, myself Sneha Gadge and today's my seminar topic is streptomycin. So friends, firstly we can see the introduction. What is the streptomycin? Then the chemical structure, their common side effects, their uses, pesticides, cell cultures, protein purification, spectrum of the activities, side effects and their mechanism. Then firstly we can see what is the history of the streptomycin. The streptomycin was the first isolated on the October in 1943 by the Albert Scats. In the streptomycin, which is an antibiotic used to treat the number of the bacterial infection. This is included in the tuberculosis, mycobacterium, avian complexes, and the endocardiasis, etc. For the active tuberculosis, it is often to give together with the isoniazide, that is rabipicin. It gives by the infection into the vein or muscles. Then we can see the structure of the streptomycin. In the structure, we can see this that is streptodine, then streptotose, and then third one it is a streptosamine, which is three groups are connected. The third one it is N-methyl L-glucose are also attached. The common side effects are include, which is feeling like the word of the spinning. First one it is a vomiting, numbness of the face, then fever and rashes. Using during the pregnancy, it may be result in a permanent deafness in the baby. Then we can use it is appears to be the safe while breastfeeding. It is not recommended in the people with the myasthenia gravis. The streptomycin it is an aminoglycoside classes of the medication. It works like the blocking the ability of the 30S ribosomal structures to make the protein which is result in a bacterial death. Then we can see the uses. So firstly, it is a medication. The effective endocarditis which is caused by the enterococcus when the organism it is not sensitive to the gentamicin. In that tuberculosis, it is a combination with the other antibiotics. For the active tuberculosis, it is often given together with the isoniazide and rafamycin. It is not the first line treatment, it accepts the medically under the served population, whereas the cost of the more expensive treatment of the probatives. It may be useful in the cases of the where the resistance to other drugs which is identified. Then we can see plague. It has been historically treated with a first line treatment. However, the streptomycin it is approved for the purpose only by the US Food and Drug Administration. It is a veterinary medicine. It is streptomycin. It is a first line antibiotic for the uses against the gram-negative bacteria in a large animals, which is horses, cattle, sheep, etc. It is commonly combined with the that is a procaine that is penicillin for the intramuscular injection. Then a tularemia infection. It has been treated with mostly with the streptomycin. Then we can see pesticides. In the streptomycin, which is also used to pesticides to be combat the growth of the bacteria beyond the human applications. The streptomycin control the bacterial disease of the certain fruits, vegetables, seeds, and ornamental crops. Then the cell culture. In the cell culture, the streptomycin it is a combination with the penicillin, which is used in the standard antibiotic cocktail to prevent the bacterial infection in the cell culture. Then we can see protein purification. When the purifying protein from an biological extract that is streptomycin sulfate is sometimes added as a it means that a removing nucleic acid, it since it binds to ribosomes and precipitated out of the solution. It is served as a method of the removing rRNA, mRNA, and even also DNA if extracted from prokaryotics. Then we can see the spectrum of the activity. Streptomycin, it can be used clinically to treat the tuberculosis in the combination with the other medication, and it is susceptible strain which causes the bacterial endocarditis, which is following represented as MIC. It is susceptibility for the a few medically significant of the microorganism. That is Mycobacterium tuberculosis, which is as a 1 mg, that is a per ml, and also 2 mg ml. Then that is a Staphylococcus aureus, that is 4 mg per ml. Then we can also see the side effects, that is a nephrotoxicity and autotoxicity. Then that is a transition of the permanent deafness. Then tinnitus, vertigo, and ataxia. Then last one, it is that is a mechanism of the actions. In the streptomycin, which is protein synthesis inhibitor, it binds the small, that is a 6S rRNA of the 30S subunits of the bacterial ribosomes, which is interfering with the binding of the formerly methionine tRNA, which is a 30S subunits. This is lead to cotton, that is a misreading, eventually inhibition of the protein synthesis, and it is ultimately death of the microbial cells through the mechanism. In also that which is Escherichia coli, which is used on it, which is surface culture that are produced, and also as in their properties, which is colorless, solid ammonia acid or base, it is a white amorphous also has a present. Then we all we can see that the streptomycins, what is the streptomycin and what they act as a, what from they arrive, we can see that. Thank you.